Welcome alumni, current students, parents, faculty, staff, and other friends of the Wiess School of Natural Sciences. I'm Tom Killian, the Dean of Natural Sciences, and it's wonderful to see so many of you here for Rice's Owl Together weekend. This afternoon, we have a wonderful program of Science in a Flash ahead of us, in which graduate students will share the excitement of their scientific discoveries with you. These junior scientists represent all of the students and faculty in the school who are seeking answers to profound questions about the origin and complexity of life and the underlying structure of the universe, and uncovering mathematical and physical laws that allow us to understand the natural world and lay the foundations for tomorrow's technological marvels. Before we start, let me give you just a few highlights of the remarkable year that we've had here at Rice and in the school. We inaugurated, as many of you know, our 8th president, Reggie DeRoche, a visionary leader who's committed to elevating our research and graduate programs, preserving our excellence in undergraduate education, and making Rice a leader in diversity, equity, inclusion, and access for students, faculty, and staff. During O Week, Rice welcomed 1,200 freshmen to campus in the second year of our expansion of the undergraduate student body. 350 of these students come intending a major in the natural sciences. The natural sciences has devoted increased resources and efforts to supporting these students, such as doubling the number of natural science divisional advisors in the colleges, and creating a new partnership between instructional teams in foundational courses and the Student Success Initiatives and the Rice Emerging Scholars programs, designed to identify students early who might need additional support, especially first-generation students and students from economically disadvantaged backgrounds. We welcomed over 400 new graduate students in the natural sciences. Among them is Nyla Davis, graduate student in mathematics, and our first Raymond L. Johnson Graduate Fellow. The Johnson Fellowship Program honors the legacy of the first African American to earn a degree from Rice, and is part of our efforts to attract the best students and increase diversity in mathematics in particular, and all of STEM fields. Students represent our future, but it's also important to acknowledge our past. So I want to acknowledge the loss this summer of one of our most beloved and accomplished professors, chemist Bob Curl. He first came to Rice in 1954 as an undergraduate, and his story is Rice's story. Humble Texas beginnings, rising to global academic prominence. He, of course, is known for the discovery of carbon-60, or Buckminster fullerene, and for which he was awarded the 1996 Nobel Prize in Chemistry, along with Sir Harry Kroto and his Rice colleague, Richard Smalley. This laid the foundation for Rice's current leadership role in nanoscience and materials research. As brilliant as he was, Bob was as renowned for his grace and his humility, and he will be greatly missed. I invite everyone to a memorial for Bob that will take place Saturday morning, November 12th, right here, with a reception to follow in Brockstein Pavilion. Bob's legacy is a guide to us as we continually seek to elevate Rice's impact and reputation. I think you'd be very proud of the talented new faculty who have joined the school in the last year, such as Samantha Uragas, Assistant Professor of Chemistry, the winner of a prestigious grant from the Cancer Prevention and Research Institute of Texas, which will support her work on the use of rare isotopes and radiolabeling to trace the metabolism of cancer therapeutics and aid in drug development. To support the growth and the ambition of the student body and our faculty, we're constructing the new O'Connor Engineering and Science Building, scheduled to be for completion this summer, which will bring together research teams to accelerate innovation on advanced materials and quantum science. We're building facilities in Tudor Fieldhouse for a joint Houston Methodist and Rice University Center for Human Performance, and an associated new exercise physiology teaching laboratory to advance research, clinical care, and educational opportunities in exercise physiology, injury prevention, and rehabilitation. Across all the programs in the natural sciences, and every aspect of our efforts to improve our world, to inspire, to educate, this is a moment of great opportunity and excitement. Scientific discovery and creating the next generation of scientific leaders has never been more important for society than it is today. For alumni, families, and other guests from the community here today, I encourage you to stay connected with the Wiess School and partner with us in these pursuits. It's been my pleasure to tell you a little about the exciting developments in the school, but it's even my greater pleasure to introduce our speakers today and to move on to our program of Science in a Flash. So we will start with Charlie Marshall. Charlie had an unconventional path to Rice. After high school, he worked on a farm, and the experience gave him appreciation for the interconnections between people and the natural world. He decided he wanted to better understand the Earth's climate and how it's changing. He earned a bachelor's degree in mathematics from the University of Chicago before coming to Rice, where he's a graduate student working with Sylvia D. in our Department of Earth, Environmental, and Planetary Sciences. Charlie is a recipient of a Kent Kennedy Institute Computational Science and Engineering Fellowship, and he successfully completed his qualifying exam earlier this week. So congratulations. Today he will tell us about climates of future past.
Thank you so much for that introduction. Hi, everyone. So I would like to start with something that might be kind of close to home for some of the people in this room. A few years ago, Hurricane Harvey hit Houston, and it was really, really bad. It was a climate catastrophe. In fact, in terms of the amount of rainfall Houston received, it was the worst flooding event in continental United States history that we have on record. So, it rained a lot today, but Hurricane Harvey was a lot worse, and we're very sure of that. I'm a paleoclimate scientist, so I try and study climates of the Earth's past, and so when we see an event like Hurricane Harvey, there are two questions we would like to answer. One is we would like to be able to contextualize events like that in terms of the record of Earth's climate variability. So in this case, it would be a question like, how rare or frequent is a storm as severe as Hurricane Harvey? Second, we would like to take that understanding of the physics and dynamics of Earth's climate that we get from studying the past and apply it to our predictions of the future. So in this case, it's a question like, under global warming, will more storms like Hurricane Harvey happen? And before I discuss the specific case, I think it's important to just go over the kind of two key tools that paleoclimate scientists use. So the first one of these are climate models. These are big computer simulations. They're basically video games with a lot more math that allow you to take what we call boundary conditions, so things like carbon dioxide levels in the atmosphere and how much energy the Earth is receiving from the sun, and then it solves a bunch of math equations for you and tells you, okay, this is the kind of climate state we would expect to see given those forcing conditions you just applied. So we really like these tools because they allow us to conduct experiments on Earth. Climate scientists are kind of restrained relative to our colleagues in the natural sciences because we don't have a second Earth we can conduct experiments on in a lab or a test tube. So we need computer simulations to be able to answer questions like, what would our climate look like today if we had not emitted any greenhouse gases at all, if the Industrial Revolution had never happened? That's not true of this Earth, so we need a different place we can do to run experiments like that. But because these are computer simulations, it's important to remember that they don't necessarily always reflect the real climates we see. And that brings me to the second tool that climate scientists like, which are these things called proxies. So unfortunately, we don't have weather satellite observations going back thousands of years because we had not invented satellites yet. And so if we want to look at climates further in the past, we need to use these chemical or biological or geological markers of past climate states to be able to reconstruct these climates. I just want to give a quick example that's kind of canonical of tree ring width. So a tree grows a ring every year, and the width of that ring depends on how hot it is and how much rain the tree gets. So then we can go and drill into the tree and measure the widths of each ring and use that to reconstruct the temperature and the rainfall at that location going back sometimes as much as 6,000 years. So on the right, this is a picture of me hugging one of the oldest trees in the world. It's a bristlecone pine, and it's very close to a tree named Methuselah, which is the oldest living thing on the planet. It's over 6,000 years old. It's really cool. And so in the case of Hurricane Harvey, we can't really use tree rings, so we tend to use sediment records. So the way these sediment records work is when a hurricane passes over the ocean, there's a lot of energy, so it tends to churn up the seafloor and move kind of coarse or large-grained sediments in ways that most other climate events can't do. So when we go and we dig, we get these sediment records, and there's a picture of one of these here. We find these layers where all of a sudden the sand is a lot coarser than it is for the rest of the layer. And we tend to interpret that as a tropical cyclone event. And so this is one of the ways we can answer the first question: how rare is an event like Hurricane Harvey? And kind of combination of modeling studies and sediment records like this allow us to say that Hurricane Harvey is a 1 in every 2,000 year event. Someone said it's a biblical storm in the sense that it's probably the worst storm we've seen since the Bible was written. So with that understanding, we can kind of better reconstruct and calculate what dynamics in the climate lead to Harvey-like events, and then we can apply that understanding to the future to answer our second question, which is, will storms like this happen more frequently under global warming? And to do this, we use a climate model, and just to briefly describe one of the challenges of this, climate model simulations of hurricanes are very difficult because on the one hand, we need to simulate basically the whole Earth because hurricanes are caused by kind of complex climate dynamics that are very large scale. So we need a low resolution model that can simulate the whole Earth kind of without using too much computing power, but we also need a high resolution to simulate the hurricane itself because the features of the storm we're interested in tend to occur on a much smaller scale. So the way around this to save computing power is to do something called dynamical downscaling, where we basically run a model with two domains, one kind of low resolution one that covers the Earth, and a much higher resolution one that just covers the storm. And so someone ran an experiment where they tried to simulate, under future global warming scenarios, 12,000 storms. So even with the kind of dynamical downscaling techniques I'm talking about, that still takes a ton of computing power, like months of computer simulations on a computing cluster, just to give you a sense of the resources required to answer a question like this. But it does let us see that as the Earth warms, sea surface temperatures get higher, an event like Hurricane Harvey goes from a 1 in every 2,000 year event to a 1 in every 100 year event. So in fact, significantly more frequent. And I just want to say that this is an example of how paleoclimate can kind of guide our understanding for a city like Houston, to say that if these storms are going to become more frequent, we're going to need to better predict and adapt to these storms, maybe not build in floodplains, and that's the value that studying past climates can bring to our understanding of the future. Thanks very much.
Thank you, Charlie. So our next speaker is Zac Spaulding. Zac decided to pursue research in algebraic geometry because of his interest in the complex machinery needed to address simple questions, such as Fermat's last theorem, which was written as a note in the ancient Greek text Arithmetica in the late 1600s. Fermat claimed to have proven that the equation x to the nth power plus y to the nth power equals z to the nth power. You didn't think you'd get a calculus lesson, did you? He claimed to have proven that it has no zero solutions when the exponent n is greater than 2. But he indicated the margin was too small to include the proof. Centuries later, in the 1990s, the theorem was finally proven using algebraic geometry as a tool. This was part of the inspiration for Zac to complete his undergraduate degree in mathematics at the University of California, Davis, and to pursue his graduate degree here at Rice, working with Tony Virilli Alvarado in our math department. Today, Zac will tell us about arithmetic geometry, turning Diophantine equations into geometric problems.
Hi, thank you for the introduction. I'm really excited to share with you all a little bit about my world of arithmetic geometry and what I do. So to start off, what is arithmetic geometry? Would be a good place to start. Arithmetic geometers are interested in posing number theoretic questions, rephrasing them to geometric ones where they can use the tools of geometry to help them answer this. So some pictures of some alpha geometric objects that arithmetic geometers like to think about. So with the vein of this, I have for us a number theoretic question that I'll pose for you. So how can you write a whole number and the sum of 3 cubes? So as far as math research questions goes, this is fairly grounded, fairly down to earth. So down to earth that it could even have been asked by the ancient Greeks. We're interested often in viewing numbers in a constructible way. So we're just going to take a look at what happens for particular values and see what the field of this question is. So for n equals 10, what are we trying to do? We're interested in finding 3 physical cubes of integer side length whose volumes sum to 10. So suggestively, you can do this. If you take one cube of side length 2 and the other 2 of side length 1, then the volumes sum to 10. 18 is a little bit more tricky, but you can do it as well. You need negative numbers. So 3 cubed plus negative 2 cubed plus negative 1 cubed is 18. Negative numbers are okay. They help us. They give us more solutions. 27 is also interesting. It itself is a cube, which gives us plenty of solutions, but there's also more interesting ones as well, such as 6 cubed plus negative 5 cubed plus negative 4 cubed. 31 and 32 are kind of special. So you don't always are guaranteed an affirmative answer to this question. Sometimes you just can't write certain numbers as the sum of 3 cubes. So for technical reasons, 31 and 32 fall into this case. And 33 is really the interesting one that I want to focus on. So, right, not too far away from all these other examples, and the answer to this question was unknown for quite some time, honestly. So this was an open question for over 60 years. Can we even write this as a sum of 3 cubes? We didn't know. We had no idea. And very recently, in the past few years, it's been proven. We know the answer. The answer is yes. And here's a small solution that they found. Each of the entries in the solution takes 16 digits, and this really is the smallest die that they got to answer this question. Right? So this is where my interest in arithmetic geometry comes, where you can ask this really simple question, this straightforward question, and the answer can be very difficult and convoluted. Right? And so what I want to focus on is like, how did we possibly get this answer? And in general, is there some systematic way for us to find out answers to questions like this? So now I'm going to go to the geometric part of arithmetic geometry and reformulate our question in a more geometric way. And I'll do so with an analogy to kind of help us along the way. Original question, how can you write 33 as a sum of 3 cubes? You might similarly ask, how can you write 25 as a sum of 2 squares? You can rephrase both of these questions as looking for an integer solution to some corresponding equation. So up top, x cubed plus y cubed plus z cubed equals 33. There exist 3 integers x, y, and z satisfying this equation. And similarly, there exist integers x and y such that x squared plus y squared is equal to 25. So on the bottom, hopefully this equation looks familiar. It's the equation of a circle. So you can reformulate this equation as a geometric object and ask, do there exist points on this circle with integer coordinates? Okay, so similarly, the equation upstairs gives for us an algebraic, starts off with algebraic information, gives us a geometric object, and we're similarly interested in, do there exist points on this geometric object with integer coordinates? So this for us is going to be an example of an algebraic surface. It's 1 dimension higher because of this extra variable, and I'll refer to this as x for the rest of the time. So I'm going to go into the land of algebraic surfaces for a little bit to just give you a feel of where I work. So algebraic surfaces, they come in a few flavors: ruled and uniruled, intermediate type, properly elliptic, and general type. And these 4 flavors are not all created equal. So rational and uniruled are by far like the most pleasant class to work with. They have a lot of nice structure that it's easy to take advantage of. Intermediate type and properly elliptic also have nice structure, but can be more difficult to write down. And general type is essentially everything else, which makes it really hard to say broad statements about surfaces landing in this class. So I focus on rational and uniruled right now. In particular, I focus on a subclass known as delta surfaces, which come in another subclass of degrees 1 through 9. And I specifically am interested in degrees 1 through 4. This is where the interesting questions happen to land. And the surface that I've given for you actually ends up falling into this chunk of classes. It's an example of the delta surface of degree 3. In the future, I hope to extend some of my work into these more interesting classes of surfaces. Back to our question, we want to know, can we write down systematically integer solutions to this equation, which can be slightly rephrased as, is it possible to parameterize the points on our surface x with integer coordinates? We want to know, if I give you some surface that looks like this, can I parameterize all these solutions in a nice way? And so unfortunately, the answer in general is no. You can't do that. This is like too much to ask for. It really is a special property to have. And so the caveat is, though, we can do something that's kind of the next best thing. So we can't parameterize the integer solutions, but we can parameterize other types of solutions that will tell us about integer solutions in general. So this is kind of as much as we can ask for and hope for. And so what are these other solutions that I'm talking about? So for all prime numbers, we have some associated number system. For the prime 2, we have something called F2, which tells us about the parity of integers, whether something is even or odd. And we have generalizations of this for 3, 5, 7, 11, and so on. They're all prime numbers. And so for each of these number systems, we have an associated surface that is related to our original x, and the surface will tell us information about the original x. So the punchline is that we're more likely able to parameterize solutions to these slightly simpler surfaces over these other number systems instead. And this will tell us about stuff for the original x. And so what have I done in my work? I've shown that if I give you an arbitrary Delta surface of degree 3, then for a 24th of all primes, we're able to nicely parameterize the solutions, the FP solutions for this associated surface. And so in very, very nice cases, you can use this data to hopefully reconstruct an integer solution to your original equation. I hope you enjoyed this little foyer into arithmetic geometry, and thank you for your time.
Thank you, Zach. Our next speaker is Tracy Yu. Tracy decided to become a chemist because, as she says, she just likes chemistry. Conducting research in chemistry gave her a sense of belonging, and knowing that her work will provide value to people in need provides her with a sense of achievement. Tracy completed her bachelor's degree in chemistry at Zhejiang Normal University in China before coming to Rice, where she works with Jeff Hartering on the path to a PhD. Tracy is a recipient of several awards from the chemistry department, including the Stephen C. Hoffman Fellowship Award for an outstanding qualifying exam, and the Harry B. Weisner Awards for outstanding leadership with graduate recruiting and for teaching. Today, Tracy will tell us about designing biomimetic materials in her talk, Building Up Proteins.
Thank you for the introduction. Hi everyone, I'm very excited to share with you my study of building up proteins. I believe most of you have played Lego. And I feel like I'm playing Lego every day, but with my molecules. The molecules that I'm interested in are amino acids, and they are like the blocks that I can connect with chemical reactions. And this will give me peptide, which I can use for protein folding. So the peptide will fold into protein tertiary structure, and then the protein structure, the Lego architecture we are trying to build. So in our body, proteins will have specific conformations, and they will require this right conformation to be fully functional. So my job is to figure out which amino acids are crucial to maintain this structure. So the protein that I'm interested in is collagen. Jennifer Aniston says, I mix the collagen powder with my morning coffee. Well, as a scientist, we tend to use collagen in a more practical way than Jennifer Aniston. Collagen is essentially the most abundant protein in our body, which is prevalent in our skins, bones, tendons. So people are always curious about mimicking the folding process of collagen to build the artificial ones that we can use for bioapplication. So what do we already know about collagen folding? People found that collagen undergoes a hierarchical folding process. If we have a peptide with the right sequence, it will have the potential to fold into a collagen tertiary structure that we call triple helix. It's essentially 3 polar peptides wind around with each other. And those tertiary structures can bundle into collagen microfiber, which can then next extend into a larger collagen fiber network, and eventually the connective tissues in our body. So my interest focuses on studying the peptide sequences that can construct these stable triple helices as well as the microfiber structure. So why do we care about this type of study? As I mentioned before, protein will require the right structure to be functional, and it turned out that a single amino acid mutation can sometimes disrupt the structure and cause diseases. And honestly, one of the most common diseases caused by collagen mutation is this osteogenesis imperfecta, which will cause brittle bones. So imagine those lovely kids born without the ability to walk or even move. We really want to know what are the secret amino acids that cause this problem and what can we do about that. So to study this type of problem, my method is quite straightforward, is to mess up the Lego scene. So suppose this is the protein that I'm going to construct. Each yellow circle represents an amino acid. I will just mess up the amino acid sequence, changing them into different ones, and see if it will cause change to my protein structure. So by messing up the blocks, it can still lead us to the protein structure, but also make the city completely tumble down. And this is what I care about, because it will tell me important information. Those blocks are important. Those amino acids are the crucial ones to maintain the protein structure. So I can chemically synthesize the peptide with different amino acid sequences, and I use them for protein folding, and I'm going to show you one of my successful folding cases. So in this case, obviously we can't see the proteins with our naked eyes, but I can see the proteins with electron microscope. So in this case, on the left side is the top and the bottom view of my protein. They look like flowers, and it turned out they are essentially collagen microfiber constructed by 6 triple helices. And on the right side, we can see these are the side views of the protein. They give us this filamentous feature that's characteristic of collagen microfiber. So this is very encouraging, but when I change the amino acid sequences, my protein structure got disrupted. And I'm going to show you some of my results. So this is a peptide sequence, and we notice that every third residue, there's one glycine. The sequence is quite repetitious. And in one of my studies, I changed the glycine to bisphosphonate, which caused my structure to be disrupted. And when we look at the model, what we can see is glycine is essentially at this pin position. And honestly, it is the smallest residue among all the 20 natural amino acids. And it will allow these 3 strands to pack really tightly into each other. So changing glycine into something larger will fail this tight packing. So this is about the tertiary triplex packing. Now I'm going to show you some results about the real protein folding. So this is a protein that's folded by 6 tertiary structures, the triple helices. So when we zoom in this bundle region, what we can see is the positively charged side chain of the amino acid that we call arginine talks to its neighbor with this negatively charged residue that we call aspartic acid. So this charge pair attraction essentially brings those bundles together to be this real protein. So those information will tell us how to design the peptide in the future that we can use for bioapplication. For example, to treat the bone fracture sites and hopefully improve the patient's condition. With that, I hope my talk gives you a quick flavor about simple molecular chemistry. Thank you.
Thank you, Tracy. Our next speaker is Danyel Cavazos. He's a physicist who studies surprising quantum mechanical effects in gases of atoms cooled to temperatures near absolute zero. As an undergraduate at Tecnológico de Monterrey in Monterrey, Mexico, Danyel received a fellowship to study abroad in Vancouver, where he was first exposed to this type of research. At Rice, he works with Randy Hewlett using physics engineering, the field of his undergraduate degree, to create complicated experiments. Danyel is a recipient of a graduate research fellowship from the National Council of Science and Technology in Mexico, and he'll be defending his PhD thesis later this semester. Today, he'll tell us about exploring the physics of one-dimensional systems using cold atoms.
Hi everyone, I'm really happy to be here sharing some of my research. So as the dean mentioned, I work in a cold atoms lab. And what that means is that I take a metal, in this case lithium, and then I cool it down to extremely low temperatures. I take it all the way up from 500 degrees to very close to absolute zero. And that means that it is no longer a solid anymore, it's a different state of matter. And you can imagine that you cannot just have it in air. I have to put it inside a vacuum chamber, and I even have to keep it floating with an electromagnet so it doesn't touch any of the walls. So this is a picture of how it looks. This is a window on the chamber, and at the center you're seeing the atoms being trapped and levitated. So I can show you a video when we turn on the machine, and it's really mesmerizing. You start catching them, and so that's how a metal looks when it's just 300 microkelvin above absolute zero. It's just really amazing to see that every day. So I mentioned that I use lithium. And you probably have a little bit of lithium in your pocket right now inside the battery of your cell phone. But the one that I use is a little bit different. It's an isotope called lithium-6, which is a little bit rare. But it's special in that it's a fermionic isotope. And that means that I can use it as a model to understand how electrons flow through metal, for example. So what I do is that I take these atoms that are very cold, and then I put them inside something called an optical lattice. So I create that by taking a laser, and then I reflect it into itself to create a standing wave pattern. But because I'm doing this in all 3 dimensions, I create a structure that is similar to an egg crate, but you have to imagine them stacked on top of each other. So you can think of this like a box that is made of light in which I can put my atoms in. And then I can take this box and change its shape. So instead of being a cube, I can make it be flat, like a pizza box, or I can make it be elongated, like a tunnel. And this will change the physics of the system. So one way you can think of it is, for example, how is it different to play in a ball pit versus play a game of pool versus using these toys? So just like each of these games has its own rules, physics in 3 dimensions is very different from the physics in a 1-dimensional world. Now it turns out most of the models that we have for understanding how electrons flow through metals assume that we live in a 3-dimensional world. And if you look around you, like that's true, right? It makes sense. Like even if you take something like a computer wire and you look at it with a magnifying glass, you can see that it looks like a cylinder rather than a line. So it's a very reasonable assumption. But as computers keep getting smaller and smaller, the wires are getting narrower and narrower. And at some point, that approximation will no longer be true. So in anticipation of that, we need to make sure that we understand the physics of 1-dimensional systems. So usually the model assumes, for example, that the excitations are individual, so they can go around each other. But in 1 dimension, that's not really possible, just because like these atoms cannot go around each other anymore. So instead of thinking of individual excitations, they become collective. One way of thinking about it, just imagine, for example, driving through a 1-lane bridge. So you have to really care about what the driver in front of you and the one behind you is doing, opposed to like driving on a highway when there's no traffic. So on my day-to-day, what I do is that I prepare these samples of atoms in 1 dimension, and then I poke it around with light, take pictures of them, and just see what they do and how they behave. They're weird, wonderfully weird, so that they pay me to just do it, and it's very funny. So one thing that we recently measured that is very different in this 1-dimensional world is that sometimes, usually when you think of a particle, it has properties like mass and charge. And when you move the particle, the properties co-move with it. But we saw, for example, when we tried to push some density waves along these chains, that the charge moved at a different speed than the magnetic properties. So, for example, when you move a chair, you will never worry that maybe you'll push the mass, but the color will be left behind. That would be crazy. But then that's exactly what we measure. So just like that, every day we get to see these very wonderfully weird things, but then we really need to make sure to understand it so that in the future, when we need to recruit this physics, we are ready for that. And with that, I'd like to thank you.
Thank you, Daniel. So our last speaker is Pedro Brandāo from Biosciences. Pedro is a graduate student in the Ecology and Evolutionary Biology program. He works in the lab of Scott Egan, monitoring the environment with molecular biology tools. Growing up in Brazil, Pedro enjoyed the natural world and has always been curious about its infinite complexity. He describes himself as a born biologist. He completed his bachelor's degree at the Federal University of Minas Gerais in Brazil and first came to Rice as a visiting exchange student before applying to the graduate program. Today, he tells a wonderful story about the biodiversity on Rice campus in his talk, Unknowns All Around Us, a new insect species on campus and its genome.
Thank you for the introduction. So hello everyone, thank you for being here. So today I'll actually not talk about something related to my thesis at all. I'll talk about a project that I had the opportunity to develop here that I think is going to be of interest to all of you. It's a new species discovered right here on campus. So species description is the most basic form of natural history. After all, acknowledging that something exists is the very first step into trying to understand it. And some species, like the Darwin finches pictured here, really require no introduction, because we all know they moved on to inspire people and change our very understanding of biology. So describing new species is not just about knowing what's around us, it's also about expanding the basis we build all of our biological knowledge on top of. So when we think of new or undescribed, unknown species, our brain usually takes us to these places like the Papua rainforest or deep sea reefs, which are really remote and not very well explored. It's true, those places are full of new species, but the new species frontier is far closer than we all realize. It's right outside of these doors, right here on campus. So I'm sure you all have learned to love these oaks that you see around campus. That is, of course, a well-known and previously described species. It's Quercus virginiana, or the southern live oak. It's been known to science since 1768. But if you look very closely at the branches of those trees, not only you will attract the suspicious looks from passerby, you also notice that they are covered in these really interesting structures. Those are called galls, and they are very diverse in their shape and form. They're caused by these really, really tiny wasps called cynipic wasps. So galls are these tumor-like structures that is a very common parasite and herbivore strategy across the natural world. Basically, when the insect or the galling organism lays their eggs in the plant, a mix of their venom and the things the larva produces will trick the plant into producing a tumor around the area they lay their eggs on. That tumor will both protect the larva inside from predators or other things trying to eat it, and also serve as food for the larva, as you can see in the picture there. And really, this can occur on most parts of the plant, on the leaves, on the stems, on the roots. But no one had quite considered if it could also be on the flowers of the live oaks. So live oaks actually have flowers. They actually have males and female flowers, but the male flowers are the ones you probably see around in the spring when they sprout and all the trees suddenly look yellow. Everyone has allergies, sneezing all the time, and they cover the ground. But when we look closely at those flowers under a microscope, we notice that they too had galls in them. So we found these really small, intricate structures that look kind of like golden little grapes, and you can see they have a little hole in them. That means that something crawled out of there. And when we looked at it in the lab, we found this very odd, yet really tiny insect crawling out of those structures. It is about 1/6 of the size of a grain of rice, so it's really small. And it was particularly abundant on this one tree on campus called the Valhalla tree, which is right across the street from Valhalla. In fact, it was so abundant on this tree, we were able to establish its complete life cycle in this tree. It's actually quite complicated, so bear with me. So the insects will find the developing catkins just before they actually erupt, and will trick the plant into producing the little tumor I just showed you, where they'll develop over the course of 3 weeks, so very fast. And the females will emerge out of those galls. The females will move on to find a place to lay their eggs. But there are no flowers anymore. The flowers only happen once a year. So what they do instead is they lay their eggs near where the base of where a leaf inserts into the branch, and that ends up producing a gall that looks completely different and almost invisible. That gall, or their crypt, or whatever you may call it, they stay there for 11 months, waiting just for the right time to emerge and lay their eggs again on the flowers. Even though scientists cannot predict when exactly oaks are going to flower each year, those bugs can predict it to the day. But as you notice, there's only ever females. Where are the males? Well, we think they might be here, but males are not that important anyway, so let's not talk about that. Well, so anyways, meet Neurotrus valhalla. It is a new species described right here on campus. It's known from its entire life cycle. I have a specimen right here I can show you later. But it's also, as you may have noticed by the name, named after the grad student bar on campus. Yes, we did that. After all, it's where we found it. And it even debuted as an unofficial mascot for Beer Bike last year for GSA. And you can see a specimen in the bar, and you go there every day, or when you go there. But what's remarkable about this is, well, we found a new species in the city, that's cool. But new species are described all the time, every day. In fact, here are some examples of species described just this year. What made this species special from a scientific standpoint is that it was also the first ever species, the first ever insect, and the second ever microorganism described alongside its genome sequence. So just over 20 years ago, we're celebrating the fact it took hundreds of people to sequence our own genome. But here we are now sequencing the genome of something we just barely discovered existed, right? And it just makes sense, because if you're describing something to science, there's no better description of a species than just provide a little recipe on how to make it, right? Which is what the genome ultimately is. So we keep learning many things. There's a lot to learn from the genome. We keep learning things about this species all the time. And in fact, every time I look at an oak, I keep finding things I don't know what they are, and no one seems to have found them before. So, you know, there might be new other species out there still. So thank you very much.
Great, thank you. Those were wonderful talks. So I hope you've enjoyed this snapshot of the research being done here in the natural sciences. I think it's fantastic to hear from the grad students for these stories. They play such a critical role in our programs, both as the mentors for undergraduates day to day working on our research projects, and teaching roles and tutoring in our classes, but they're also essential to their creativity. Their hard work is essential for the research for which Rice is known. So I encourage you all to stay, and please come and talk to these amazing students and ask them more about their talks. Enjoy the food and the company. And on your way out, please grab a t-shirt for Natural Sciences so you can take it away and remember your association here. So thank you very much. Let's thank our speakers.